May I invite Dr. Jayaram and Dr. Dhruv Mamtura will chair this session.
So, good evening, everybody. So, Dr. Jayaram Iyengar, firstly, he needs no introduction. I think everybody knows him, but still, I'll introduce him. So, Dr. Iyengar is MBBS from Osmania University, Hyderabad, and MD Pathology from PGI Chandigarh since 1987. He's silver medalist. Also, he's a winner of Excellence in Teaching Awards from JNMC Belgavi, and he's a winner of several national awards. He's a member of various national and international associations and has presented many associations of India, and he's a member of accreditation committee to NABL and chief faculty for Foundation of Quality and program coordinator for NewCap EQUAS program and has over 42 publications. So he has also delivered over 250 lectures. So we are adding one more to that. So welcome, sir.
Thank you very much and a very good afternoon to you all. I think I am the odd person out in this session because perhaps mine is the only non-hematology talk, and in these 15 minutes I'm going to talk about something that all of you already know. I'm going to talk to you about policies which are statements of intent and addresses the question what, and of course objectives which are means by which the policies are fulfilled. And these objectives could be multiple, which are directed to achieving whatever is stated in the policy. These need to be measurable, they need to be achievable and aligned with the policy. And the talk of mine today is about the quality indicators, which are nothing but analysis of the data which is obtained when all these objectives are carried out and they are fulfilled, to know whether we are in line with what we set out to achieve and whether the entire purpose is satisfactory or not, and the path for continual improvement is set once these quality indicators are diligently measured. So we'll try to understand very basically, very macro level, we'll understand the what and the why and the how of quality indicators or key performance indicators. And in the current context, everything is risk-based. The ISO standards have moved from process-based to risk-based activities. So let's try to see how we need to realign our quality indicators towards the risk-based approach. The quality indicators, I will not go through the definition. Quality indicators will measure how well an organization meets the needs and requirement of the users as well as the quality of all the operation processes. Why do we need to define and why do we need to measure, analyze these quality indicators, especially in the context of modern laboratories? We tend to achieve more with less. So the providing high quality results rapidly, providing high quality results with minimum expenses seems to be the goal of each one of us in the audience. And very small inefficiencies in the entire process can take up the costs. They can prove to be very expensive. And in this context, quality indicators play a very important role in the improvement process. Improvement not only financially, but improvement in the overall quality of the laboratory. When we actually set out to select quality indicators that we need to monitor, and we would probably make a big list of quality indicators that we would want to monitor, and somewhere we get lost in the entire, the volume of the data which is going to come in, the number of parameters that we are going to analyze. So certain key points we need to remember when we need to select quality indicators are the needs of the customer. Are we focusing our quality indicators to meet the requirements of your customer, to meet the requirements of your society? We have an obligation to the society. And of course, our own weak points, our strength, when we do a SWOT analysis, our own weak points, the vulnerable areas in the laboratory need to be focused, and you need to develop quality indicators towards that. And then these quality indicators will also depend on the resources that are available in the lab to achieve these indicators, to monitor the indicators, obtain the data, and so on. And of course, all quality indicators need to be aligned with the policy or the mission of the organization. Now, broadly, if you look at these, some of the quality indicators are required by law, by regulation, accrediting organizations, and contractual requirements. Now, most of the other quality indicators are chosen by the leadership or the management of the lab based on the organization goals, based on risk, based on strategic planning, review of data of whatever we have had, as well as review of the complaints, again gives us the areas that we need to improve upon, client feedback, and also subjective assessment. Some of these quality indicators are, again, the management should take a broad view of the operations, considering known risks and potential consequences. So we are trying to align ourselves to a risk-based approach to the quality indicator, entire analysis, and so on. And on a broad scale, I think every aspect of laboratory services should be objectively and regularly monitored at some point of time. And if you look at the CLSI guidelines, there are about 12 the quality system essentials that are defined, and we need to focus on various of these areas and shuffle them to see that all of these get monitored at some point or the other. But then when we select quality indicators on a priority basis, we need to consider a few of these points. One is this a critical examination? If it is critical, that takes high priority. If it is a very complex activity with many inputs, and a classic example is turnaround time. Everybody talks about turnaround time. Actually, believe me, monitoring turnaround time is a very complex process because there are so many inputs which are involved, so many timestamps that are involved. And getting an ideal data analytics of the turnaround time is always going to be a very complex task, so that takes priority. Everybody requires the reports on time, so we need to analyze that. Then if a process is highly automated or is it a manually driven process, that may help you choose certain quality indicators. Then our own weaknesses. Is there something that is vulnerable and known for repetitive problems? In my own lab, I know that day in and day out, we have patient registration problems, the demographic errors as something which is repetitive. So are we monitoring those things? And then if we've already brought in certain improvements and we need to monitor to see whether those improvements are effective, that could be one area where you select certain quality indicators. So broadly, if you look at pre-examination, examination, post-examination, your quality indicator should span across all the three processes. And we need to apply risk management tools to narrow down this huge list of quality indicators that we've listed out to those which are more meaningful, and in that current context, they are the more important ones. That is what we need to focus upon. And all these quality indicators, I keep repeating, should align with the organization goals. So the key points here is do not select too many quality indicators to monitor at a given time. You may list out a lot of them, but select the few which are very important based on your risk assessment. Do not chase wasteful quality indicators which are not going to give you any meaningful information. And if you find that a quality indicator is stable, do not chase that stable quality indicator. Do not ignore it completely, but do not actually focus upon it with all your, the resources and so on. Now, I don't know if you can read this. Just to explain, there are a few publications which have kind of developed certain harmonized quality indicators, which makes life relatively easy for laboratories. So they have developed a whole list of harmonized quality indicators, which are in the pre-examination, examination, and the post-examination areas. Now, it does not mean that every lab has to take all these harmonized indicators and start monitoring them. Now, in a given activity, among the list of harmonized indicators, one lab may choose a few of them, again, depending on how frequently those are contributing to errors or what is the impact of that error on patient safety. So if you look at one example, I think this is out of focus. If you look at one example, the inadequate volume of collection was found to be one of the causes for a pre-examination error. Among the list of 3 or 4 potential indicators, that was found to be more often found. So that was monitored by the laboratory and they brought in the necessary corrective measures. So that's what this entire fishbone diagram wants to achieve, that whatever the list is there, you pick out those which are relevant to you. So indicators broadly can be of broad categories. I think this we have already covered. They cover critical processes, they cover quality system requirement, or those that are mandated by accreditation bodies. What is important is selection of quality indicators with care so that time and effort put into them is not wasted. The whole list of indicators which are there, all of these need not be monitored at a given point of time. So once you are identifying a quality indicator, we need to develop the quality indicator monitoring process, for which you take one of these, each one of these, and then you define the purpose of that quality indicator. So identify the quality indicator, identify the scope, identify the purpose, bring in the authority, bring in the list of people who are authorized to monitor these indicators, and which domain or which department needs to be defined. So this gets into a formal kind of a documented process. Once that is done, data needs to be collected, data needs to be analyzed, and you need to develop a strategy for analysis and collection of data, because ultimately data collection is the key to a good quality indicator process. So you collect garbage, you're going to get garbage out. So good data collection is the key. So you need to have a documented process for data collection. You need to bring in responsibilities, define frequencies of monitoring, define the source of data and the type of data. These are very critical things which give a good quality indicator monitoring. And what is the scheme of sampling? How do you sample the various things? Then setting targets is very important, because you need to look at a goal, you need to work towards that goal, and the limits and action threshold. So if it exceeds that target, what is the action we are supposed to take needs to be defined. And this will be based on current performance, will be based on references, and these targets should be feasible, they should be achievable. Let's not be idealistic and set very high targets that we may not achieve. That demotivates the staff. Set targets that can be achieved. I'll run you through some examples of, actually these are actually being done in some lab recently which I had audited, and the indicator here they've taken is the amended report. What do they say? The objective is to monitor frequency of the amended report. So that is very clear statement. What is the purpose and what is the rationale? The amended reports need to be communicated to the patient, because treatment can change if the reports are amended. Now, how data is collected is defined. What is the measure is also defined, percentage of amended reports. The frequency of monitoring is defined and benchmark is defined. The targets are defined. So this is how very clearly the purpose and the process is defined. Similarly, if you look at CBC report turnaround time for emergency lab and stat reports, similar kind of a process is followed. Very clearly, the people are aware of what needs to be done, how it needs to be done, who is going to do it, at what frequency it should be done, and so on. And what is the acceptable target. So once this is done, it is always good practice to do a trial run, do a short pilot study and review the results of that trial run. And then once you're satisfied, launch a full-fledged program. And collection of indicator data is very, very important. Once this data is collected, this data needs to be represented, needs to be interpreted and analyzed. And once it is analyzed, the data needs to be presented. And this data, once it is presented to the management will be able to take various decisions based on what is the outcome of the quality indicator data. So if you look at this, the tabular column of sample rejection, how many samples were rejected, percentage of samples rejected, listed out. So these are all listed out. A benchmark is also defined, and this is always a better appreciated thing, that you have a graphic representation of the quality indicator data. But the story did not end there with this lab. Even though they have achieved the target, the lesser number of rejected samples compared to what is their benchmark, still they split it. And they found that insufficient samples exceeded a benchmark, exceeded the cap benchmark. Similarly, they found that leaked tubes exceeded the benchmark. And then they tried to analyze it further to see from which of the collection points were these more often seen. And then they start plugging them. So that way, if you do that kind of analysis on a micro level, you can target the potential areas of problems and you sort them out. And then action plan, no action plan required. So what would be the action plans required? The responses to an outlier in the quality indicator performance should be actionable and involve planning and implementing for change and improvement. What is important here we should remember is you need not bring about dramatic changes in your system. Even small changes done in increments can achieve a lot over a period of time. So you may decide to continue monitoring, you may decide to stop monitoring that particular indicator, you may identify opportunities for improvement, you may bring in corrections, corrective actions based on root cause analysis. Again, a proper root cause analysis, I think most of us do not do a proper root cause analysis. Once you know the exact cause for that problem by asking the question why multiple times and arrive at the root cause, you target the root cause, you're going to plug that problem. So that is something we need to develop and develop an entire quality management strategy. You may need to modify the targets, modify the threshold. If we've been consistently achieving a target of 5%, you may want to tighten it and make it 3%. That takes your improvement levels to a higher strata. And then, if necessary, report to the appropriate parties, customers, accreditation bodies, public, and so on. So that needs to be decided. So the take-home message at this point is that quality indicators are an integral part of the quality management system with all the ISO requirements and the CLSI documents. Development of quality indicators should be based on both risk assessment of potential errors and the frequency of observed errors, and the risk of error and likelihood of harm is minimized using quality indicators. So if we follow a risk-based approach, our entire outcome is going to be safer to the patient. We are not going to cause harm, we are not going to cause injuries to the patient. So a risk-based approach to quality indicators makes the outcome more meaningful and safe. So this, my friends, is a very broad approach of how we need to rethink about the way in which we are defining and monitoring quality indicators and bring in the element of prioritizing your quality indicators based on the outcome, based on the risk, based on the harm or potential harm that we are likely to cause if we do not monitor those processes. So, thank you very much.
I thank you, doctor. So, any questions in the audience?
What is the difference of the quality indicators of the key performance indicators, the KPI?
KPI and
Quality indicators is
I
the
think
same?
they should mean the same because quality indicator and a performance indicator, they both measure the same thing where whether you're achieving what you set out to achieve and is that achievement meeting your targets or not.
Yeah. And in the Mayo Clinic in Rochester, Minnesota, I have learned the quality system, and every 2 years they were reviewing every process to decrease, to improve these quality indicators showing the number of reject. Do you propose any certain time to improve every method in the lab to have better quality indicators?
I think it would not be wise to make a definite frequency with which these reviews need to happen. It all depends on the laboratory. Now, if a laboratory has developed a quality system and it's young, they may need to review these more frequently. Whereas a mature organization, they start reviewing it at less frequent intervals. But what is important is do not monitor these quality indicators just to provide some data for the assessment when the assessors come to look at your lab. Do it for your own benefit. Do it for the benefit of your patients. And that's where you realize that many quality indicators which you feel are not required, you just keep them aside. Take the more important ones.
And you use the Pareto rule to sort the problems, for example, for rejection of samples, to focus in the 20% of causes that produce the 80% of the rejections?
I think we could use a lot of these data.
Okay,
Statistically.
thank you so much.